All right, so this is the introduction to Lab 3.2, Endospore and Hypothesis Testing Lab. What I'm going to do today is I'm going to be boiling some samples of different species of Bacillus and also E. coli, and we are going to test your hypothesis. So in the pre-lab, I won't give a page number because it could change, but the pre-lab you should form a hypothesis. A hypothesis is a proposed explanation for a natural phenomena. So a good hypothesis should be written to limit the number of variables. So this is a pretty simple, straightforward lab. I'm simply going to be boiling test tubes of the bacteria by placing it into a water bath here and taking periodic samples. What we're testing for is the ability of the endospore to actually withstand the high temperature from the boiling water. So certain gram-positive bacteria will form endospores. The two clinically significant genera of bacteria that form endospores are Clostridium and Bacillus. They're considered clinically significant because there's species within those genera that can cause disease. So Clostridium botulinum that causes botulism, Clostridium perfringens that causes gangrene, Clostridium tetani that causes tetanus, and then one that you may be familiar with is Clostridium difficile, which causes antibiotic-associated diarrhea. Other organisms from the genus Bacillus, such as Bacillus anthracis that causes anthrax, and Bacillus cereus that causes food poisoning, would be other clinically significant bacteria capable of forming endospores. One of the common misconceptions is that all organisms that are gram-positive form endospores, and that is not true. There are other gram-positive bacteria that form endospores, but the two clinically significant are the Clostridium and Bacillus. So why would a bacteria form an endospore? Clostridium and Bacillus are found in the soil. A soil can be considered an extreme environment because during the day in the heat of the sun, the soil temperatures can get really hot. Bacteria in the soil can go through periods of time where there's very low nutrients and very low moisture. So a bacteria capable of forming an endospore will do so when the nutrients are depleted in the environment, or maybe the cell density is too high. So I like to give the analogy of considering yourself a bacteria and you jumping into a swimming pool, and that swimming pool is full of cheeseburgers, and there are only so many cheeseburgers in that swimming pool for you to eat, and each time you eat one of those cheeseburgers, you multiply and divide yourself into another one of yourself. So a binary fission process. Now you can imagine that space is limited and that food is limited in that swimming pool. So eventually the space in that pool is going to become crowded and the number of cheeseburgers is going to be decreased. So you'll be competing with yourself for more food. So what some bacteria do that are capable of forming endospores is they go through a process of sporulation. And that sporulation process takes about 8 hours. So when the nutrients become limited and the cell density becomes high, the bacteria will trigger the sporulation process. Sporulation is the process of forming the endospore. Once the bacteria is in the endospore state, that bacteria is dormant, it does not metabolize, it does not reproduce, it simply is a dormant structure. And while it's in that dormant structure state, the endospore, they tend to be highly resistant to things like heat, resistant to chemicals, and resistant to desiccation. So they're not metabolizing, they don't need the water, so they're resistant to those things. They're also resistant to UV light. By placing the bacteria in the boiling water, we're testing the ability of the endospore to withstand the heat. These bacterial samples have been incubating for 1 week, so the nutrients are very limited and the cells should have depleted most of the nutrients in there, and endospores would have already been formed in here. So an endospore forming bacteria will go through a cycle beginning with a vegetative cell. The vegetative cell is the active metabolizing cell. Once the nutrients become depleted or the cell density is high, they go through that process of sporulation. They are then in the endospore state, and they are in the resistant dormant state. Once optimal conditions are met, so you take that endospore and you put it into an environment with nutrients and the right temperature and some moisture, that bacteria will germinate and form back into a vegetative cell. And it's the vegetative cells that cause the disease. So they're clinically significant because A, some of them cause disease, and B, they can withstand common disinfection procedures using things like heat and chemicals and UV light. So we're using E. coli. E. coli is a gram-negative. This is kind of going to be our control. We're going to place these into the boiling water at the same time. I have Bacillus cereus. Bacillus cereus is commonly causes food poisoning, so usually associated with buffets. I don't have any Clostridium because Clostridiums are usually anaerobic, and we don't have the capabilities of growing anaerobes here in the lab. So the first thing I'm going to do is I'm going to take a time zero and actually plate the bacteria onto the media to see what it looks like growing before we put in the boiling water as a control. So I'm going to get my loop red hot to be aseptic. I'm going to pick up the Bacillus cereus, resuspend this. You can see there's a big chunk at the bottom there. Pass that through the flame. Get a loop full of the Bacillus. And I'm going to put it on my plate, the spot labeled time zero, just a little streak of the bacteria. I'm going to repeat that for all of the samples, and then we have the last one, the E. coli. So you should have formed a hypothesis for the E. coli in heat and the Bacillus in heat. Propose an explanation. What do you think will happen after 1 minute, after 2 minutes, after 5 minutes of the bacteria being exposed to the boiling water? So I'm going to place the Bacillus cereus in the water, and I'm going to set a timer for 1 minute and then take it out and take a sample. Alright, so it's been 1 minute. I'm going to put this on the time 1 spot on the plate. This is this tryptic soy auger. And I'm going to place the tube back into the boiling water for 1 more minute and then take another sample. I'm going to repeat this at 5 minutes and at 15 minutes and at 20 minutes. So it's a little bit different than what's written in your lab manual. And I will repeat that for all of the samples. We will see what will happen with E. coli. So that's 2 minutes for Bacillus cereus. And I'll place that back in the boiling water. I now have E. coli going at the same time. It will sit in there for 1 minute, another minute, for 2 minutes total, 3 minutes total, 4 minutes total, and 5 minutes total. So I'm going to do E. coli a little bit different just because I know E. coli doesn't really like the heat. Some things that you should be able to answer or explain would, number 1, be the endospore cycle, like I mentioned, using the terms vegetative cell, sporulation, germination, what causes sporulation, what endospores are resistant to, and then here's something to consider. When I place Bacillus in the boiling water, does that actually trigger sporulation? When do the endospores actually form? Okay, so think about that. That's a question in your lab report. So I have E. coli finished here. E. coli was boiling for a total of 5 minutes, and I still have the Bacillus cereus going. It will go for a total of 20 minutes. I will take a sample here at time 15. I've got 2.5 minutes left before I need to take that sample, and then I will take a sample at 20 minutes for that as well. When I sample, when I take that out of the boiling water and I take a sample, and if I observe growth on the TSA plate, What does that mean? Are the vegetative cells that are in here growing on that TSA? Or is it the endospores germinating? So think about that as we wait and then observe the results here in the next few minutes. All right, so we're going to look at the lab report for lab 3.2, look at the results, and we're going to change some of these times from 3 to 5 minutes total, 15, and then 20 minutes. And what you'll do is record pluses for the amount of growth. And we are also going to use Bacillus cereus as our organism to begin with. So here we have our time 0, our time 1 minute, time 2, time 5, 15, and 20. So you can see that there's growth for each one. Time 5 doesn't have as much growth, but that's basically due to when you put the loop in, how many of those endospores you're actually grabbing. So here I didn't really get much, but you can see that Bacillus cereus has a really resistant endospore that it forms. So even after 20 minutes in the boiling water, we have growth. So when did the sporulation process begin? Did the boiling water trigger that? Or were those endospores formed before the experiment even started? So think about that, and think about the endospore cycle and how long the sporulation process starts. So question number 8 in the lab report: when Bacillus is placed in the boiling water, does that trigger sporulation? When do you think the endospores were formed? And again, think how long the sporulation process is and what causes bacteria that can form endospores to actually form the endospores. E. coli. So here we just went from 1, 2, 3, we'll do /4 minutes and 5 minutes. And here we can see that E. coli, we have our time 0, and after 1 minute in the boiling water, it's dead. So the vegetative cells are quickly killed by the boiling temperatures. E. coli does not form endospores, so it is quickly killed by the boiling temperatures. Question number 9. When I sampled Bacillus from the test tube that was in the boiling water and plated the sample on this media, explain what is happening if you observe growth. Am I sampling vegetative cells? Or are the vegetative cells killed? Am I sampling the endospores? And did the endospores then germinate? So what is happening? Again, comparing to E. coli, E. coli does not form endospores, so those vegetative cells are quickly killed in the boiling water. Freeze-thaw. Here we have Bacillus subtilis, we have E. coli, and you can see that even after freezing and then thawing, both of them have growth. So freezing is a method of preservation. It is not killing the bacteria. So even though Bacillus forms endospores, E. coli does not, the freezing has no effect on the ability to grow. So that concludes the Lab 3.2 video. Hopefully this helps you get a better understanding of endospores and their significance clinically that can impact your job as a healthcare professional.